I'm Julie Wolf of IndieBio, and I'm joined today by Damien Kudella to share his founder story. Damien is CEO of Cayuga Biotech, with a drug candidate called Cy1 to change the way that major bleeding episodes are treated. Thanks for joining me today, Damien.
Thank you for having me.
Now, Cayuga Biotech was spun out of the discoveries that you made as a graduate student. Can you tell me why you decided to, instead of taking an academic route to continue studying the discoveries that you had made on your route to PhD, decided to go into an entrepreneurial route?
Mm-hmm. So I had actually never really envisioned an academic route. By the time I was done, I enjoy science, but the research lab part of it, I was a little burned out on. And so I was looking for a new way to apply my science knowledge outside of the lab. And so specifically, the interest was kind of seeded by a conversation that I had in DC about my 4th year of my PhD. I was thinking about, what I wanted to do because I didn't want to be a professor. And the person said, this technology is really interesting. We have a real need for it. And, you should think about doing a company to advance this technology. So we had already been in the early stages of patenting my thesis, the Cy1 drug. And so I figured, I've already been a starving grad student. Why not go be a starving entrepreneur as well?
If you're accustomed to scraping by, why not continue along that path for just a little while longer?
Just a couple extra years.
Yeah. Was there anything about the clotting cascade or bleeding specifically that you were interested in studying?
My interest is more about how we can take science and use it to help people. Obviously, in this case, it's medicine for saving lives. I will say, ironically, my biology is probably the weakest, certainly of our 3 key members out of Kyle, Nate, and myself. But it was really, nanotechnology is an interesting field, and we could take a lot of what we had learned from biology, in this case, polyphosphate as a polymer, and really apply it and make it work better. And so I think that's really the next stage of a lot of drug development is whether it's oncology with kind of more targeted delivery, in this case with lowering the payload and coming up with new ways to target the clotting cascade. I think that's hopefully going to be the next big step.
Let's dig in just a little bit to the biology here and the polyphosphate, which is produced normally within in the body by platelets. Can you give a brief overview of how platelets and polyphosphate work together to promote clotting?
Sure. If you think about a clot as a brick and mortar, platelets form the brick, then there's this compound called fibrin that's the mortar, and so that's the physical clot. And the problem really becomes how you get to that brick wall that plugs the wound. And so polyphosphate's job is it's produced by platelets, and it's essentially a catalyst. So I'm a chemist by training, so catalyst makes the most sense to me. But its job is really to speed things up. So you put polyphosphate in there, and the clotting cascade that's ongoing, it goes quicker. And that also enables you to shut it off quicker, which actually has some safety benefits as well. So let's say your patient, not only are you saving their life by reducing blood loss, but you can actually reduce the time it takes for them to heal as well.
I see. And so catalyst, in my mind, is something that speeds things up, as you said. And the action of this polyphosphate, the Cy1 drug candidate, is a little bit different than other drugs that are out there right now to treat bleeding episodes, right? How do they differ?
Typically right now, a lot of the drugs are recombinant factors. They're either direct mimics of endogenous proteins or slight alterations. And so the issue there is if you think about the clotting and thrombosis as sort of a seesaw, so typically you're flat and then your patient gets injured, they start bleeding, that gets expressed. And then unfortunately what can happen with the recombinant drugs is that you get the balance out of whack and you take a patient who's like this, and now there's what's called throwing clots. And so that's a really big problem. All the drugs right now have a black box label warning because of that. And so a doctor really has to weigh, is my patient critical enough that I'm going to take the safety risk? And so what we've designed with polyphosphate, sort of being a catalyst, you should have, again, we're having an effect on the rate, but we're not having an effect on the specific factor. So for example, polyphosphate has its biggest effect on the factor thrombin, and we're not actually producing more thrombin. So sort of, this threat that it's going to lead to clots, sort of a gray goo kind of thing. That's not going to happen. And also because it works within the body, the body inactivates thrombin very quickly to ensure that clots remain only where they're needed.
I see. So there's an inherent difference in safety profile when you're basically making something natural happen faster versus increasing the amount of material within the body, which is what some of these other drugs do. Is that correct?
Exactly. And because polyphosphate has a more limited effect, let's say, than a clotting factor, you can sort of use it hopefully in a safer way.
And I know your team has done some animal trials to look at safety, looking for the presence of clots, which can occur naturally just by the body, but also in the presence of some of these other drugs that may add to the amount of material within the body. What have you seen in terms of clots that are showing up, let's say, in lungs or brain or other parts of the body?
Mm-hmm. So we've been looking at different tissues, specifically lungs are huge, especially when you're talking about nanoparticles. The pulmonary beds, that seems to be where a lot of nano-based drugs fail. So that was a huge concern. Obviously, the liver, since that plays such a huge role in clotting, and then everyone, of course, the blood-brain barrier. So what we've done on our own is we've compared it to saline in our pig model. And what we've seen is that there is no difference between saline or the Chi-1 drug. And so, like you said, pigs are very hypercoagulable. So there was some evidence of clotting, but it was in both the saline and the Chi-1 drug. So that is likely a result of sort of the pig's natural clotting cascade as opposed to Chi-1 causing any safety issues. So obviously, safety is the number one concern, especially in this field. The first question we're always asked is it safe? Is it effective is always number two. But we're on the pathway, and we believe that everything that we've seen so far and will lead us through, safety, both the remaining IND enabling studies as well as our clinical trials.
And so when we're talking about bleeding and clotting, this is a really delicate balance. And I think a lot of people intuitively could understand how a drug to treat bleeding would be necessary in the case of traumatic bleeding, where there's a lot of obvious bleeding. But there are other places, other situations, and one could say patient populations that could benefit from Chi-1. Who else do you think will benefit from Chi-1 coming to market?
Mm-hmm. And you're right, that's actually kind of the story of Cayuga in my PhD thesis. To date, we had been, until IndieBio, we were fully funded by DARPA and the Army. Obviously, it's a huge problem on the battlefield, cause of about 50% of deaths. But in the US, there's also a huge problem. So especially as patients get older, and they may, they end up on anti-clotting drugs. So things like Plavix, Coumadin, some Xarelto, and some of these newer drugs. So the problem, like you said, these patients are kind of already in the clotting phase. You give them the anti-clotting drug and they go back to bleeding. And a lot of it is done because there's no treatment. On top of that, there's also, we've been focusing more recently on platelet dysfunction. So this could also benefit patients on chemotherapy who end up with thrombocytopenia, as well as patients who have congenital platelet disorders. So we have identified a hemophilia-like genetic disease that affects platelets as opposed to factors 8 or 9. So there's a wide range of people, and obviously everybody thinks of quantity of life because bleeding can kill very quickly, but it also enables these patients to have a better quality of life as well. You don't have to worry that shaving or an accident in the kitchen can be fatal. And so it really, you can help give these patients their life back.
Absolutely, that's an excellent point. Now, you mentioned earlier that you have some co-founders who are also PhD scientists. Can you tell me how you and Nate and Kyle came together to form Cayuga Biotech?
Sure. So about fourth, fifth year as I was finishing my PhD thesis, data was really good on the benchtop and we were figuring what to do. And so I was at UC Santa Barbara, and the sole graduate student who really does any animal experiment at UC Santa Barbara at the time was Kyle Pluntz. So Kyle actually has a very good story of the first time we met. I'll let him tell it another time. But I ended up working with Kyle. You know, we have this new drug, we want to test it, and we hit it off while we were doing the test. And you know, data worked really well. We got along well together, and so Kyle and I jokingly refer to that Cayuga is our baby. So
The two
we
parents.
decided, yeah, so we decided we were the two initial co-founders, and then in sort of 2019, so we had been a contractor on an Army SBIR grant. And so we needed to add a third person to do a lot of our sort of quality control as we're working on our manufacturing. And so Kyle was a postdoc at UC Santa Barbara, and we figured, you know, Kyle, you know of anyone who's going to be a good fit for Cayuga? And he said, Nate. You know, we had a couple of quick interviews, thought that Nate was excellent, and brought him on board. And so it's been, you know, really excellent. So really, we're all tied by UCSB. I think I was the first one to get the PhD. Kyle's a little bit later, and then Nate had to struggle the least. So a little jealous of him.
Nice. That's the banana slugs, right? UC Santa Barbara?
Gauchos, actually.
Gouges. Oh, I see.
Yes.
Nice. Well, I'm glad that your road led you to meet each other and join us at IndieBio. Now, the road is going to be a long one for a drug that needs to get approved through the FDA and undergo clinical trials. What is the next major milestone that you anticipate Cayuga will make after the IndieBio program? And where do you see the company going in the next, let's say, 5 years?
Mm-hmm. So we've already had our first meeting with the FDA. That was in May. And so we've gotten feedback. So really what we need to do is finish up our PK, PD, and tox studies. So
Which means, I'm sorry to interrupt you. I just want to explain that a little bit. PK/PD, that's kind of a complex term. What's that mean exactly?
So it's pharmacokinetic and pharmacodynamic. Give a very generalized. It's how the drug works, how it's cleared, toxicity. You know, are there any adverse effects from the drug? Obviously, you mentioned previously, we're very worried about clotting just because of the field we're in. But really, you know, what doses should be effective in an animal model, what doses are safe. Hopefully that's the same thing.
A whole bunch of questions, right? All in one. All in
exactly.
One phrase, PK/PD, a lot of questions, right?
Exactly. Yeah. And so really determining that it's safe enough to move to human trials. Mm. So that's our next big thing. Thanks to IndieBio, Kyle and I were able to get back into the lab and run some more tests on a variety of different bleeding disorders. And so we're hoping to have all of our data completed and an FDA approval to start clinical trials at the end of 2021. So let's say 5 years will take us out to 2025. You know, typically drug development takes about 8 to 10 years in the clinic. In this field, because it's such an unmet need, there's room to move it quicker. So the FDA has its what is called the Accelerated Approval Program. And so we're hoping to use that, you know, ideally, we if the drug is as safe as everyone at Cayuga believes it is, we think we could actually be approved for use by 2025.
Wow, that's great. Well, I know that you're quite busy trying to make that happen. So I'd like to thank you so much for taking time to speak with me today, and good luck to you as you continue in your IndieBio and company journey.
Thank you. Glad to be here.
Great.